Click for IB Biology. Here we're looking at the DNA replication mechanism required on the higher level course. There's a PDF of this presentation down in the description, which you can annotate with your own notes, and there you'll also find a 10-question quiz with answers. Links to the channel and to other videos will follow at the end of this video, and in the description you'll also find an AI prompt that you can use as the basis of your own research. So let's look at the ways in which the higher level model differs from the standard level model. So as before, we have the DNA molecule shown here as the double helix, and as previously mentioned, students do not need to be able to draw the double helix, but you are expected to be able to draw what is known as the ladder diagram, as shown here. And this is actually just a small section of the paired nucleotides, the anti-parallel chains, so showing the 5' to 3' going in this direction, and then the anti-parallel polynucleotide going in the other direction. The complementary base pairs are in the center here, A to T, G to C, T to A, and of course C to G. Helicase is in position and is opening up the hydrogen bonds as previously described, so that either side of the DNA molecule can move apart to act as a template for replication. But here's the first additional point for your learning, which is that this replication actually occurs in what are called replication bubbles, and there are numerous replication bubbles along the entire DNA molecule. And in those bubbles we have helicase operating at this end and at this end. So the helicase would be operating in this direction and then operating in this direction, a pattern which is repeated along the entire DNA molecule. And in so doing, this makes the replication of the molecule much more efficient. It would simply be faster. And so here we have again the representation of the DNA replication. This section of the model has already been explained to you, and we can see the helicase here progressing up into the DNA molecule, separating the hydrogen bonds to separate the polynucleotide chains, and each polynucleotide chain is going to act as a template for the replication of the entire molecule. In this representation of the process, we have the helicase in this position working its way up into the DNA molecule. The position of the helicase enzyme on the DNA molecule is called the replication fork. And so one of those is shown in this diagram here. And on this side, where the 5' is in close proximity to the helicase, then this has 5' to 3', and we refer to the original polynucleotide chain here as the leading template strand. Over here we have the other original polynucleotide, and where the 3' is in proximity to the helicase, this is known as the lagging strand template. So leading strand and lagging strand. Then what happens, of course, is that we have the nucleotides will come in and complementary base pair with the lagging and leading strands. And this introduces our second new feature at the higher level, which is that if we take the leading strand as our first example, the initial nucleotides that will add to the polynucleotide and complementary base pair here are not DNA nucleotides, but are actually RNA nucleotides. And these are referred to also as primers. These RNA nucleotides, which are added to the chains, are added by the enzyme DNA Primase, which by some authors is known also as RNA primers, RNA primase, and you can regard those two enzymes as being the same thing. Now, in my model, I only have 4 pairs of nucleotides. In the real situation, the number of RNA nucleotides added to the beginning is somewhere between 8 and 12 base pairs. So here I've just represented that with a single one. Now this has consequences. So for instance, if our leading strand begins with adenine, then the complementary base pair here would be uracil and not thymine. So we have to remember that the RNA, whilst DNA has the bases, DNA has the bases A, T, G, and C, RNA nucleotides would come in with A, U, G, and C. And so we have this change here of thymine to uracil, which I've illustrated here. The nucleotides are adding to the beginning of the chain here 1, 2, 3, and 4. And you'll notice that I've added my RNA nucleotide at a higher position on the lagging strand. This complication will be our third point, which I will present in the next video, part 2 of DNA replication mechanism. The link to that video is appearing in front of you now, along with the link to the playlist and, of course, to the channel.